Okay, let's go ahead and get started. I'm Suzanne Warrenfeldt. I'm part of the outreach team here, and I'm joined by my colleague Omar Harb, who is the director of outreach. We also, Sarah Kelly should be joining us from London to answer help questions behind the scenes. We hope to see her soon. And we are here to fill you in on some new features and data from Release 55 and to demonstrate how to access and interrogate some of that data. You can ask questions as we demo things. In your GoToMeeting controls, you should have a questions panel. And if you have a question during the webinar, you can type it in there and we'll try to answer it as soon as possible. And sometimes we can stop for discussion also. Okay, so I will go to VEuPathDB. So this webinar is being recorded and you will receive an email tomorrow for a link to the recording. All of the upcoming and recorded webinars appear on the webinars page, and I'm showing the VEuPathDB site here, which is our portal to access data from all organisms that we support. To get to the webinar page, I'll go to the help section, learn how to use VEuPathDB, and click on this webinars button here. If you don't get the email or if you can't find it, then you should be able to find it on this page. And then one other thing to note is that all of our sites are designed the same. So if I go to AmoebaDB, the home page looks the same. All of these menus contain the same information, but the underlying data is different. It addresses the organisms that are grouped into this AmoebaDB site. So everything that we talk about today is available on any of our sites if that data type is available for those organisms. So to begin, I have a few overview slides about the release, and I will go back to my presentation. So in release 55, actually, it was released on the 2nd of December. It included 62 data sets. And those were integrated into 9 of our projects, and they included 7 different data types. So we could see FungiDB received 20 data sets, host, micro, and pure PlasmaDB all received 1 data set each, and vector-based and PlasmaDB also received quite a bit of data, totaling about 62 data sets. There were 7 new data types, or the 62 data sets that we integrated spanned about 7 data types. There were 27 data sets that had new or updated genomes or annotation, 1 chip, 1 chip seq. Our vector-based map view tool received 8 new data sets. And we had some proteomics, RNA sequencing, and isolate resequencing data also. And each of these different data types addresses a different biological feature, depending on the technique. There were 4 new features that we integrated into all of our databases. We have a new tool called the My Organism Preferences, which I will demonstrate here in a little bit. And it's an option for filtering the menus of each of our databases into a subset of organisms, just to make the interface a little bit more manageable if you're only interested in a few things. We changed the color scheme, or are offering a new color scheme for our reference sequence track that should help people visualize that reference sequences and start and stops for different genes. We updated our Galaxy version on October 8th. And this Galaxy version, well, Galaxy in general is a platform for analyzing your own data. And we offer ways to port your analyzed data into VEuPathDB to analyze it further, privately. But this version of Galaxy improves the job and workflow performance. We've also added some tools that you can check out there if you're a Galaxy user. And then we've also enhanced the searching on our record pages from the content bar on the gene page. Our gene pages have so much data that they can actually take a long time to render in the browser. So by default, we keep most of those tables closed. But we have a tool in the content section, a filter, that allows you to access the table descriptions. So you can search from the content section to look for data on the page. Okay, so I'm going to now I would like to demonstrate my organism preferences. Oh, and here we are at AmoebaDB. Let me make that a little bit bigger. So my organism preferences is a way to filter all of these menus for the searches and everything to the organisms that you're interested in. So let's say I'm going to click on my organism preferences here. And let's say I am only interested in 3 different organisms. I can expand all, clear all, and then I can tell my organism preferences that I'm interested in Acanthamoeba castellani Neff. The other one I'm interested in is Inventimeda invadens. And let's choose another one, the Fowleri. I can say apply that. And now I see that my organism preferences includes 3 of 28 organisms in the database. And if I go to these different searches, my organism parameter only shows me 3 organisms. I can turn that on and off very, very easily. I can just say disable, and then all 14 of the organisms are available, and I can run this search based on that. Now, that's interesting also on the RNA-seq to look at the functional data. I can look at all of the data available or only those for the organisms that I'm interested in. Okay, so I think that is what I wanted to accomplish with my overview of the features. And so I'm going to hand this off to Omar, who, will start looking at the data a little bit closer. And Omar is going to describe about half of the data, and then I'll come back and tell you about PlasmaDB and VectorBase.
Okay, sorry, I was having trouble finding the mute button. So let me switch over here and share my screen. So hopefully everybody can see my screen. Suzanne, can you confirm?
Yes.
Okay, perfect. All right, thanks, Suzanne, for the overview. And one thing I wanted to stress or maybe rehash again from what Suzanne said, this my organism preferences parameter is quite useful, especially when you come to our resources that have many more genomes. So for example, I'm looking here at the VEuPathDB portal site, and you'll see here that there are 573 genomes available, which is a daunting number, and you probably only care about a handful of them. So you can easily go in here and really set your parameters based on, for example, a clade. So I could go in and say, well, I just want to select, apicomplexa, for example, which include organisms like malaria and toxoplasma, plasmodium and toxoplasma, and then oomycetes, which are also alveolates, and you may want to compare them somehow, and you can apply this. And so all of a sudden you're limiting, you're filtering the entire site based on these organisms, which allows you then to focus your work and only see data sets of interest, as Suzanne described. So I won't go into that further. So I'm going to cover a few key data sets that we loaded. There are many data sets, as Suzanne showed you, and we're definitely not going to cover all of them. It'll take too much time. So I'm going to just take you on a tour of a few databases quickly, show you some of the data sets that were loaded, and for some of them I will take you into an in-depth demonstration. And as Suzanne indicated, if you have any questions, please use the question panel in your GoToWebinar control panel. You can type any questions there. We'll monitor them. Both of us, Suzanne and I, and also Sarah Kelly is online as well, and she'll be answering questions in the back end. So either we'll answer them by typing the answer, or we'll answer them live if it makes sense. So looking at FungiDB, again, with any of our sites, let me go back to the homepage. We have a news section, which is typically collapsed by default using tweets. So we highlight different things in our tweet thread. But if you go to the news section, you can go to the latest news, for example, for release 55. You can quickly see what is new in FungiDB. And as you can see, we've loaded a number of genomes, sequence, and annotation. And so you can go in and search for genes based on different characteristics, like whether a gene is called a kinase, or whether it has a predicted signature peptide, or it has transmembrane domain. So different types of searches you can apply to the different genomes. We also load genome sequences. So for example, you'll see here there are 2 genome sequences without annotation. And those are where an assembly was generated, but annotation was not generated. Those are still useful because it allows you to blast against those sequences. And in fact, we provide a blast against NRDB, which allows you to find genes. And I'll show you an example of this in TrickDB, not in FungiDB, but we'll so hang tight to see that demonstration. We also loaded a number of population biology data sets, specifically data sets that did whole genome sequencing of isolates, and then determined whether these isolates have polymorphisms based on, and of course, those are quite useful for you to then identify regions of the genome or genes that have specific types of polymorphisms based on the isolates that were studied. For example, if you're looking at a human pathogen, then you could imagine isolates that came from a blood sample versus on your skin, and you want to compare and find if there are any things that distinguish these isolates from each other based on their SNPs, or whether something is drug resistant or sensitive, and so forth. And I'll give you an example of this in a second. We loaded a couple of RNA sequence data sets. One of them was for Clavispora lusitaniae, which is this is actually a candida. This is the telomorph stage of the organism. I will show you or demonstrate this RNA-seq data set. We also loaded a data set from Aspergillus fumigatus, and for each of these data sets, as I'll show you in a second, we have a description page, and so you don't have to just rely on this little sentence here, and I'll show you that in a second. Now, we also loaded some chip data, and we've also included updates to the annotation based on manual curation, and so there are 208 genes from Aspergillus fumigatus that were updated, and you can click on these links and examine them further if you're interested in knowing what those specific changes were. Now, hopefully what you can appreciate is that there's a variety of different data types that we can load, and this is only a sampling of it. And so as you think about the experiments that you're running in your lab or experiments that you read about in papers. And if you notice anything that you think is missing from the databases and could be useful to the community, we'd love to hear from you. That's obviously, there's no way we can scour the literature and attend every conference in the world to find out which data sets are being generated. So I encourage you to contact us. As you can see, we can handle all sorts of data types and amounts of data as well. Okay, so I'm going to quickly go to this Clavispora data set, and I'm going to click on the data set name right here. You'll see it's a hyperlink. This will take me to what we call a data set page. And so this page includes a number of sections, including a top section here, which gives you a quick access to the publication. So this will link to PubMed. And then from there, you can, in many cases, get free access to the paper. We have information about the primary contact for this study. So in many cases, people would like to contact the person who generated the data. And we highly encourage that. We don't generate this data. We're simply giving you a way to access the data and ask questions about it, as I will show you in a second. We provide a description here that should give you sufficient information to understand how you may want to search this data. So for example, in this case, this study looked at a number of isolates from the same patient, and these isolates were either fluconazole resistant, labeled as U, or fluconazole sensitive, labeled with the letter L, okay? And those are coming from a single clinical sample. Some description about the method, and then as we scroll down here, we provide you, all the way in the bottom here, we provide you an example of what this data set looks like in the database. So you can quickly see here that, oh, here's a couple of L samples labeled with L. So these are the sensitive strains, and these U ones are the resistant strains. And you can see this particular gene, CLUG03087, which hopefully doesn't mean anything to you. It's just a gene ID, but this gene has this particular expression profile in these different strains that came from the same patient. So this is all well and good, and you can go and visit specific genes and look at their expression profiles for a specific gene. But we also provide you ways to search this data. And so you'll see here in the section, we have a section called Explore This Data in this website. And it provides the different ways where this data is presented or available for searches. And you will notice here that they are the RNA-seq searches. We have a fold change search, we have a differential expression search, we have a percentile search, and we have a antisense transcript search. In this case, I'm going to look at the differential expression search. So this means that this data set had biological replicates, so we can actually calculate a standard deviation, essentially, and look at the expression profile. And what you see here is that with this differential expression, you can compare two samples to each other, right? And this is our sites remember your last search. So this was actually the last search. I was trying this out right before the webinar, making sure that everything works. But so what I did here is I selected this L10 isolate, comparing it to this U5 isolate. And I'm looking for genes that are upregulated. I'm looking for them to be upregulated by at least twofold, and I kept the p-value at its standard. But you can change this, of course. And now, as soon as you click on Get Answer, this will return results. So these are 43 genes that met the criteria of interest. So there are 43 genes that are upregulated in one isolate versus the other. Okay. I think I picked the resistant versus the sensitive isolates. Anyway, that doesn't really matter which ones I ended up picking. The bottom line is I have two different conditions. I compared them. I have these genes that are differentially regulated. You can see here that there's this gene here is a multi-drug resistant gene, which is differentially regulated, upregulated in the samples I selected compared to each other. And in this case, since we're looking at a resistance versus sensitive strains, getting a multi-drug resistant gene in your results is a positive thing. Okay. All right. So what I'm going to do here is I'm going to expand this strategy quickly. I'm going to add a step. And I'm going to ask another question about these samples. So these are, remember, these are coming from this Canada species. And in this case, what I'm interested in doing is I want to actually leverage data from other organisms in the database. Okay, so what I'm going to do is I'm going to add a step, and I'm going to convert these 43 genes into the orthologs or the homologs of these genes in Canada albicans, because I know Canada albicans has, we have a huge number of isolates that were sequenced, and we called SNPs on those isolates, so we can actually now ask, by orthology, whether the orthologs have a specific types of SNPs in them. So I'm going to go ahead and transform by orthology, and let's hear. So I could, if I want to, I can expand this tree and try and find Canada albicans as this tree, which becomes, again, very unwieldy. There are many organisms in the database. Or I can use this filter quickly, and I can just start typing albicans, and you'll notice here that it filters my tree, it prunes it for me, and now I'm gonna select the reference in this case and run my step. So this tells me that my 43 genes from this species here on the left has 61 orthologs or homologs in Canada albicans. Okay, and very quickly looking at the names of the genes in Canada albicans, you'll see like this first one up here is involved in fluconazole resistance somehow. So that's again a good sign, and there's a multidrug again, which also is a good sign that at least the search is doing what is expected of it. So now I want to ask if these 61 genes have any polymorphisms in there, and I can actually be very specific about the type of polymorphism and the number of them. So I'm going to add a step here, and I'm going to look for, so I'm going to intersect the results from my previous search, which is the 61 genes, with the results from a new search that I'm going to conduct right now. And the new search is going to be a SNP search or a polymorphism search, and you can type filter or terms in this filter window or filter box, and it'll filter your searches for you. I find one now called SNP characteristics. Let's check this out and see what it has. Starting at the top, you can see that there's an organism drop-down menu. So I'll look in here, and remember, I transform my orthologs into Canada albicans, so I'll stick with Canada albicans here. And now what you'll notice once I select Canada albicans, this table here updates and provides now the information for the isolates we have for Canada albicans. And you notice here that we have 255 isolates from Canada albicans. We have information about these isolates which are categorized here on the left. And then the details of these categories are presented here in the middle section. And so you can imagine a sample type. We can look at collection year. You can look at the geographic location. So for example, if you said, well, I really want to only look at isolates that came from France for whatever reason, or maybe you know that there's a study that was conducted in France and that's the one you want to pick. Or alternatively, you can also just go to the study, the data set itself, and now you can see the study names. And I can say, well, I know this study here that collected 182 isolates from Canada albican isolates from different parts of the world is a good study. So I'm going to go ahead and pick these 182 isolates. All right, so remember right now, all I'm doing is I am setting up my SNP search, my variant search of the Canada albicans data we have in our database. I chose 182 isolates to compare. There are a number of parameters here, which it would take us about half an hour to go through them in detail, but we do have a lot of help for each of these parameters. And we have a number of tutorials that go in depth into how you can play around with this search. So each of these can give you additional ways to interact with to specify the types of SNPs you're interested in. For my purposes, I'm going to go ahead and pick only non-synonymous SNPs. I'm going to ask that these SNPs are present in most of the isolates, so 80% of the isolates that I selected. So 80% of these 182 isolates must have a SNP at the position that's returned, basically. So this allows you to increase or decrease the stringency of your search. And I'm going to ask for something crazy, like 20 non-synonymous SNPs. So now I'm asking for any gene in Canada albicans based on these 182 isolates that have at least 20 non-synonymous SNPs in them. And I'm going to intersect the results with my previous search right here in the background, which is the 61 genes that are upregulated in fluconazole resistant organisms. And I'm going to run the search. So the result may be 0, and that's totally fine, right? The cool thing about this is that this is an in silico experiment that you can go back and modify. You haven't wasted any lab reagents. So you may look at this and say, if you get 0 results, you can go back and you can modify it and reduce the stringency to see if you get back results. In my case here, there are a total of 452 genes that met my criteria. It seems like a high number, but there are about 452 genes that have at least 20 non-synonymous SNPs in them from most of the isolates that were in that study. 5 of them are also part of my 51 or 61 genes that are orthologs of my fluconazole resistant strains. Okay, and so now you may be interested in looking at these further, because this may indicate that these genes with these high number of non-synonymous SNPs may, the locations of those SNPs may be critical for the fluconazole resistance as well. Okay, and so that'll be one way where you can explore the data and delve into it further. Okay, so let me pause for a second, see if there are any questions. Suzanne, anything that needs to be answered on air, or have they been taken care of?
No, we did have a question about our SNP pipeline, or analyzing the whole genome sequencing data. And so I put a link to our analysis methods, which gives an overview. And I also pointed out that our Galaxy instance has a pre-configured workflow that
Great.
folks
Thank
can
you.
use on their own data. Yeah.
Perfect. Okay, so now I'm going to quickly jump to TrickDB. And so TrichomondisDB, TrickDB, we'll look at the latest release. And you'll see here that we added a new genome, Trichomonas fetus. And so again, this genome you can explore further. You can learn more about the genome, where it came from. And then there are a number of searches that allows you to explore this data. So very quickly, I can, for example, look for genes that have a predicted signal peptide. Let's say I'm only interested in genes that are secreted or potentially secreted in Trichomonas. So I can add this. I get 1,027 genes. I can ask, well, how many of these genes also have a transmembrane domain, for example? So let's go ahead and look for transmembrane domains. And so we'll select that. And not only that, I'm going to say, well, how many of them have at least 4 transmembrane domains? So I'm making the minimum transmembrane to 4. And remember, I'm intersecting. So I have one search here on the left, which is my signal peptide positive genes. And the new search is looking for any gene that has at least 4 transmembrane domains. And I'm going to intersect the results between these two searches. And you'll see here now that out of 1,818 genes with transmembrane domains, 73 of them also have a signal peptide. And so then you can go back and look at them and explore them further. I wanted to highlight, we have, as you remember, I mentioned to you that we not only integrate annotated genomes, like this one is an annotated genome, but we also integrate unannotated genomes. And I just wanted to quickly highlight a way where you can explore an unannotated genome effectively. So I'm just going to go to the tools, go to the genome browser. And the genome browser, if you go from the tools, it'll default to whatever default organism we have. And in this case, it's Trichomonas vaginalis. But I'm going to switch. So I'm going to click on this genomes menu at the top left-hand side of the genome browser, or JBrowse in this case. And I'm going to pick Trichomonas tenax. And so this is a species of Trichomonas that is available without annotation. But we have other types of data that you can load on here, which I already preloaded for you. And one of them, which I think is quite useful, is this NRDB protein alignment option. And this is basically doing a blast, basically, between the assembly itself and NRDB. And so anywhere you see a little line there, a little graphic, is indication of whether there is homology there to something in the NR database at GenBank. And so now you can quickly click on these. So for example, I'll just click on one of these, and you'll see this is homologous to a hypothetical gene in Trichomonas vaginalis. But without even annotating this genome, you can imagine where all the genes are on this particular contig, right? So if you were working on this organism and you really wanted to know where certain genes are, you can load this track, and then as you zoom in, you can zoom in and look at the sequence itself, which will appear, and you have to zoom into a certain point, and it'll appear right here. Okay, and then you can move around until you find a gene, for example, and you can explore it further. Okay, and now quickly, I know I don't want to take too much time. I know Suzanne has a bunch more things to cover. So looking at TriTriDB, you'll see here that again, we've loaded a number of genomes. So there are a number of Leishmania species that were loaded. These are pretty nice, actually, very, very well assembled, high throughput third generation sequencing. So these are all PacBio and assembled. They look like pretty nice assemblies. And so it's a nice way then to use these for cross-species comparison from various different species that we have in the database. We've also loaded a ChIP-seq data set. And so again, this is another type of data that we've made available to you. I don't think I'm going to highlight anything in here in TriTriDB since I highlighted these in other databases. Just to point out again that we do have some active curation and annotation from our curation team, and this indicates what has been updated in the last release. I'll take a quick tangent and indicate that we have different ways for the community to provide annotation to us, new or updates to the annotation. One of them is through user comments on gene pages. So if I go to, let's see here, I'm just going to pick a random gene in FungiDB, and you'll notice here that there's an opportunity to add a user comment on this gene. So for example, if I knew that this gene, because of the work I've been doing, has a, if I knock it out, is essential, right? So I can go in here and add a comment and just, you have to log in. And then once you log in, you can add a comment and give that information. Or you can maybe say, well, this gene is not a putative membrane protein, it's actually a soluble kinase, right? So you can provide that information. And then for genomes where we have support for active curation, we get annotators look at that and actually use that to inform the annotation. But regardless of that, your contribution is essentially crowdsourcing annotation. And we have thousands of annotations or community comments in the database, and I would highly encourage that. Another tool I'll quickly indicate to you is the Pollo annotation platform. And again, you would have to log in, but I would encourage you to go to this page. So if you're interested in providing structural annotation of genes, and this is something you really enjoy doing and you have information about this, I would encourage you to go to the Pollo annotation platform. And this allows you to then provide direct knowledge to the community, because any changes in Pollo will appear on gene pages automatically. And if we get a chance, I'll show an example of that. So quickly hopping on to ToxoDB, just to highlight that we loaded an Eimeria tenella genome. This again is a really good assembly and worth looking at for cross-species and evolutionary comparison. We also loaded a palmitoylated proteomic data set, which again, you can go in and search it and look for genes that have increase in palmitylation based on the experiment that was done. I won't highlight anything here, just suffice it to say that, again, explore this data, and if you have any questions, you can contact us anytime. I'll quickly go to PlasmaDB. So PlasmaDB includes Theileria and Babesia organisms, and we've loaded a transcriptome during development for Babesia bovis. And so we can take a look at this and learn more about this data set, which publications are associated with it, some description of the data set. So you'll see here that this was RNA isolated from Babesia bovis that came from ticks or bovine blood. So quickly, I would be interested in asking, well, are there any genes that are upregulated in bovine blood compared to tick I'll pick upregulated. I'm gonna select 20 as my fold difference. So I'm saying I want to find genes that are upregulated by at least 20-fold in bovine blood compared to ticks, and with a p-value of 0.005. So I'm being very, very stringent here with my search criteria. This results in 163 genes, which you can take a look at and, of course, try and explore further. One way that I will highlight quickly before switching back to Suzanne is that you can also, when you have a list like 160 genes, you can look at them manually one by one and try and determine if there's something in there that's interesting. But you'll never know if what you're looking at is really statistically significant, or are you just anecdotally looking at that and thinking, oh, I feel like there are more transporters in my list, right? So you'd like to get something statistical. And so we have an option for you to analyze your results. This is really highly used, actually, and there are different ways to look for enrichment in your data set. So in this case, I'm going to go ahead and use a Go enrichment, gene ontology enrichment in my 163 genes. So I'm asking if there's any function that is in Go that is enriched in my 163 genes compared to the entire genome. So is there anything in here that happened that is not that did not occur by random? So I'm going to pick biological process, and let's see what happens if I select that. I'll run this, and you look here, and it looks like there are not many numbers here. And I'll look here at the right at my p-values, and these p-values are not terribly good, right? So based on this in the biological process, there's no enrichment. But you can go down and change these. Look, well, is there any enrichment in Go terms for cellular component or molecular function? So I'll go ahead and select molecular function. And this is a nice example where you can get enrichment based on what you're looking at. And so you'll see here at my top one, protein binding, there are 14 genes in my result list compared to 339 in the genome. But statistically, this is a significant enrichment, which indicates that these 14 probably did not occur by random, right, in my gene list. So that's pretty cool. So that means that there are some 14 genes that I may be interested in looking at, and those may define genes that are upregulated in the blood stage versus the tick stage of Babesia. So we can look at those 14 genes. If I look at this table, you'll notice here it says 14, and it's hyperlinked. So I can click on that, and this is going to take me to a new strategy that has these 14 genes. And now I can look at them, and you'll see that they're all SMORPs, right? So these are all antigenetically variable surface genes in Babesia, which are expressed in the blood stage. So this makes sense. So no surprises here. But the cool thing about this is that it shows you how you can play around with these different data sets that are published, explore them, mine them again and again, and potentially find new features or interesting genes that you may be interested in exploring and looking at. So Suzanne, I'm going to stop right now, unless there was anything else you wanted me to cover before I switch it back to you.
No, that sounds good to me. I can go on to PlasmaDB.
Okay, perfect. So I'll make you the presenter.
Okay. Do you guys see PlasmaDB?
Yes. Mm-hmm.
On screen. Okay. So for PlasmaDB in release 55, there was quite a bit of data, and let's go to the news release and review that. So we had a genome sequence, so an unannotated assembly, and then we had 4 new annotated genomes added. We had 5 or 6 RNA sequencing data sets, 3 or 4 proteomics data sets, and this first one, the ubiquitome, during the asexual stages, we have a host component of that data too. So when the authors collected their samples, they also collected the host proteins and analyzed those to define the ubiquitome during infection with P. falciparum. And so we have that host component of the data on HostDB. So not only can you interrogate the data here, the parasite version, but also the host ubiquitome there on HostDB. And then further down, we see that our curators have been hard at work updating the annotation of many genomes. So they review papers and possibly new RNA-seq data to add new functional annotation to the annotations that we represent. And so if you want to delve a little bit further into that, you can download these GFF files, but then we also create a Google page that gives an overview and then chronicles each change that was integrated during this release. So that's good. So for a demo, I'd like to show you the p-falciparum data that is a 48-hour time point, a 48-hour time course transcriptome at 2-hour resolution. It's a paper that was published in Nature last year, and it defines a circadian rhythm for p-falciparum. I will, instead of going to the dataset record page, I'm going to go to these searches and go to the RNA-seq page just to show you a different way to get there. So I'm at searches, search for genes based on transcriptomics and RNA sequence data. And then you can see that we have many data sets integrated for the Plasmodium species. But the one I want to look for is this new one. And I'm going to use the fold change search to look at, to interrogate this data and pull out groups of genes. So you can imagine that with this very detailed data set, 2-hour time points at over 48 hours, you can imagine that groups of genes will have different profiles across these 48 hours. Some genes will have a high expression very early on in the life cycle and a low expression later on in the life cycle. So we can use this search to pull out groups of genes with certain profiles. And so let's go ahead and do that. I'm going to look for upregulated genes that are at least 5-fold different, looking for upregulated genes in the later time points of the first life cycle that they recorded. So 20, 22, 24, sure, 26, and compare that to the expression of genes in the early time points. Okay, so I'm going to click Get Answer. And I find 2,599 genes that are with this profile. And if I can, of course, make that more stringent. If I would like to, I can go back and revise and say I want genes that that are 10-fold difference between the end of a life cycle stage and the beginning. I could also, change both of these to average to look for a more median type of expression change, and I come up with 1,673. And if I can, and then I can just look at the data that we have, made available for you, right? So indeed, all of the genes that have been returned have very low expression in the early hours and high expression at the late hours of the first life cycle that they've recorded. So that's pretty cool. And we can, as Omar demonstrated, we can cross-reference these 1,600 genes with any of the other type of data that we have using our search strategy system. But let's say, we can also download that so that we have our list of genes that we can save forever. Let's go ahead and edit this and revise it and change the strategy a little bit for what we want to do. Say that we want to look for genes that are upregulated in the beginning of the life cycle and have lower expression in the at the end of the life cycle. I'm going to reverse my choices and say that for the reference samples, I want to do 20, 24, and 26, and clear all of these and say our comparison samples are 0, 2, and 4. So now I'm looking for genes that are upregulated by a factor of 10 in the early hours of the life cycle stage versus the of the life cycle versus the end of the life cycle. I'll say revise, and I end up with 197 genes. And indeed, we can go down here and look at our data. These are genes that are highly expressed at 0, 2, and 4, and then at the end of the first life cycle, they are down. So it's a pretty powerful tool to pull out different profiles, especially with a data set that is so intense and has evenly spaced transcriptome data. Okay. So now I will turn our attention to VectorBase. And go to the news there. VectorBase received quite a bit of data this go round too. We have genome sequence and annotation. We have one new genome and then another genome, the Foshan FPA version of ADs received some annotation updates where they have renamed many of the added stable IDs to this organism, so that might be something that you want to look at. And then for 4 organisms, the community annotations have been integrated into the from Apollo into the official annotation. We also have several RNA sequence transcriptomes that were integrated, and all of these are really fairly new from 2021 on down to 2018. Several proteomics data sets, and then some population biology too. I'd like to talk about our Apollo community annotations, and then a little bit about our population biology or map view. So 4 organisms received Apollo updates, and Apollo updates, as Omar explained, is a community annotation platform that we support. And in Apollo, a user can make suggestions for improvements to a gene annotation or a gene model, and we will incorporate that into the official annotation after it's reviewed. So these 4 genomes received enough annotation and attention from the community to warrant an update in the database. And when we do that, we increase the annotation version number from, by 1. So for example, Anopheles quadriannulatus went from .1.2 to .1.3. So let's consider this genome. And so here I am at the record page for Anopheles quadriannulatus, and I see that it was actually published in 2015. And as Omar pointed out, you can search this data as you like. But the thing about it is that since 2015, there may have been experiments, other data available that help define gene function or alternative gene models that, data that was not available in 2015. So Apollo allows users to enter that data and so that everybody can take advantage of it. And I'll show you just a quick overview of what Apollo is. This is a sort of just a landing page, but it has a lot of help information if you're interested in delving deeper into Apollo and adding more annotations. So here we are at Apollo. I will change the organism to quadriannulatus. So what I'm looking at is data tracks on the left, and on the right are my controls. and I would like to search for a specific gene. Say I'm studying this gene. Okay. Right, so you could use this filter up here to look for a certain gene. If I click on that, it's going to take me right to it. And then I can turn on some tracks. I want to make sure that my draggable tracks are on. Have annotation and this analysis track that's going to show me. We've actually analyzed all of the RNA-seq data in our in vector-based DB and come up with a track that says that shows alternate introns and alternate exon spacing based on the RNA sequence data. So we can turn that on, and then anytime you want to make a change, all of these tracks are draggable, and you should be able to use these draggable tracks to create different alternate transcripts, and then you can save that. Our curators will see this, they'll review it, and when each genome has received enough annotation modifications or additions from our community, then we can integrate that, and those will appear in our official annotation track. So for the take-home point is that for release 55 is that these 4 organisms have enough updates from the community that we integrated that into our annotation. So that's great. And let's go back to the news. And we can see that our population biology, we integrated 7 projects and updated 2. These data are generated in field studies of mosquito surveillance. So I will go to map view so we can get an overview of what it looks like. So this data is mosquito surveillance. So somebody sets up a trap, they capture mosquitoes, they take them back to the lab, they count them, they do see what the distribution of species from their collection point are, and then they possibly do some genetic analysis on them. So they might look for microsatellites or different allele changes, different mutations in each of those. And then we integrate that into this tool so you can delve deeper into each study, but you can also relate all of these studies to each other. So that is available here. You can, we have several tutorials online so that you can learn how to use this a little bit more. And then the other thing that we've done with for this population biology data is that we've actually integrated all of the metadata into the site so that each of these studies now has its own dataset record page. So I'm going to go to data, dataset records, or datasets in VEuPath VectorBase. I will search the category field for population biology. And I see that I have 708 data sets out of 1,209 data sets showing. So for each of these data sets, we have an actual dot on the map view tool, and you can go to these data set records to learn more about that study. Yeah, so we've made a lot of progress in providing the metadata associated with these data. I think that's all I wanted to show. Did you have anything to add, Omar?
No, this is excellent. So I think the last thing maybe to point out to everybody is definitely if you have any questions, drop us a line. We have a pretty active contact us link. So we get emails regularly on a weekly basis, but also feel free to use the chat box at the bottom right-hand side of any of our sites. It's a community chat, so we can answer questions there, or other community members can answer questions there. And I think that's it. So thanks everybody for joining, and we will see you next time. Let me see if there are any questions here.
I think they're mostly answered there.
Sam provided a link to the contact us if anybody wants that. And I think we answered any of the all the questions. We can hang on for one more minute in case anybody wants to ask a question, since we're a couple of minutes early. Yeah, I don't see any additional questions, Suzanne. So I think we're good to end the webinar. Thanks everybody, okay. And we'll see you next time. Bye-bye.
Y'all take care. Bye.